Hello, everyone, and welcome to today's webinar, a single-cell and spatially resolved atlas of the human and murine oral cavity. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Akoya Biosciences. To learn more about our sponsor, please visit akoyabio.com. Now, today's presentation is interactive, and we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Now, without further delay, I would like to introduce today's speaker, Dr. Inês Sequeira, Senior Lecturer in Oral and Skin Biology, Deputy Director of Research, Institute of Dentistry at Queen Mary University of London. For a complete biography on our speaker, please click on the Presenter tab at the left of your screen. Dr. Sequeira, welcome. It's such a pleasure to have you today. You may now begin your presentation.
Hi, good morning or good afternoon, everyone. I'll start by sharing my screen here with you. Right. It's a pleasure for me to present you the work that we've been developing in my lab. My lab is at Queen Mary University in London, and thank you so much for the invitation to present. And today we will going to be presenting you the work that our lab has been developing in spatial biology, particularly our work in establishing a human cell atlas for the oral cavity and a mouse cell atlas for this tissue as well, encompassing both single-cell technologies and spatial biology. On a broad vision, my lab is interested in basically understanding the interactions between the fibroblasts, the epithelial cells, and the immune cells. And you have a very interesting projects being developed in the lab looking at the role of keratins in immunoregulation and in carcinogenesis, looking at tumor heterogeneity in oral cancer. But today I'm going to focus in two projects: our international effort to create the human cell atlas and how my lab is contributing to the oral and craniofacial atlas, and our goal to understand the outstanding potential of scarless healing that the oral mucosa possess when comparing to skin. So for those that are not familiar, in the Human Cell Atlas, we are actually setting out the goal to create a reference map of all the human cells, basically like creating a Google Maps of the human body. And our ultimate goal is basically to understand what are the common and the unique cell types that are supporting the different niches in health, and uncover which cell types and networks would then be affected in the case of disease. So while this is the first atlas, we'll be mostly focusing in healthy individuals from adult tissue. We have a growing effort in sampling distinct ethnicities and ancestry background at different stages of life, and also being able to understand how this impacts the disease severity, the outcome, and ultimately also the treatment. Just put the pointer here because it's probably easier for you guys to see. Perfect. Right. So within the Human Cell Atlas, there are several bionetworks for each organ, and in the oral and craniofacial that I'm coordinating together with Kevin Birds at the American Dental Association in the US, and we have the support also from champions from different institutions that are contributing not only with data sets, but also in the crowd annotation that we have for the different tissues, making sure that we are taking into account the diversity of the different tissues. So the ultimate goal is basically to map all the niches of the oral cavity in health and then compare those with disease. And the driving forces pushing this endeavor at the moment are Bruno Matuk, Diana, and Lin Zhang. Bruno and Diana in my lab and Kevin's, and Zhang at the Sanger Institute with Sarah Tachman. And they've been working in the challenging task of integrating multimodal data sets that have been generated. And at the moment, we are finalizing the first version of a single spatial and a single cell, and a spatial human cell atlas. And this is the work I'm presenting you today. And if you want to know more about our project, you can read the vision of this project in the roadmap that we published a couple of years ago. So as you may be aware, there are multiple niches within the oral cavity, including the labial and buccal mucosa, the major and the minor salivary glands, the gingiva, the tooth, the tongue, the hard and the soft palate, and the tonsils. And all these tissues are supported by different types of epithelium, stromal and sensorial, vascular, and resident immune cells. And this actually reflects the different roles of these different tissues that assist us in talking, in breathing and eating, masticating our food, tasting it, and even kissing. So, however, what happens is there is a very limited knowledge on the diversity of the cell types, the cell-cell communication relationships, and their spatial patterning, comparing between different niches. So we are using a multimodal approach to uncover this heterogeneity in the oral cavity. So for that, we're following a workflow which starts with the integration and annotation of different published and newly generated single-cell RNA sequencing datasets. And at the moment, we are using mostly 10x single-cell and single-nuclei RNA sequencing datasets. And after our own filtering and QC pipelines, we are using the Selenics platform from Bioimage for basically be able to have a user-friendly platform crowd annotation of the datasets. And this is really important because we want to make sure that we have a consensus annotations, as I was mentioning before. Then, to study how cells communicate with each other, we use multi-niche-net that allows the analysis of the cell-cell communication within multi-sample and multi-condition. So in our case, addresses the inter-niche heterogeneity. But of course, as you all know, single cell requires the dissociation of the tissues into single cells, and all the spatial information is lost. That's where the spatial biology then plays a crucial role. So not only we validate our findings from the single cell analysis, but also to add the spatial information, we use the PhenoCycler-Fusion platform from Akoya Biosciences, which is basically a multiplex immunofluorescent platform allowing you to use several markers, to 100 markers at the same time on one single slide. So in this project, we use a 44 antibody panel with the main structural markers and an extensive list of the immune markers, together with some custom antibodies that are important for our tissues, such as, for example, special keratins that are expressed in the oral cavity. And to also on the spatial field, but now on the transcriptomic level, we use MERFISH from VisGen with a panel of 300 probes. And the advantage of these techniques comparing to other methods is that you can have the single molecule resolution, allowing you to pinpoint each transcript to a single cell. And so we were then able to combine spatial proteomics, and transcriptomics from serial sections for downstream analysis. And as you can see, this is a big collaborative effort that combines cutting-edge techniques and really needed the development of new bioinformatic tools. But most important, and basically the central feature of our project vision, is that this atlas is available on an open access online portal for all the scientific but also clinical community to be able to explore. Now focusing on the actual data. So we were, to define the cellular heterogeneity across the different niches of the oral cavity, we integrated 14 published and newly generated single-cell RNA sequencing datasets from adult health oral cavity with over 70 samples from 14 different studies covering the 13 different niches that I described. And in our atlas, we also have representation of both genders, age from 18 to 69-year-old, and different ethnic backgrounds as well. And after reanalyzing and filtering all the data sets with the same pipelines, and this was done at the Sanger, these data sets were then uploaded on the Selenics platform, resulting in an atlas of almost 250,000 cells. And we started by doing this first on a broad basic cell annotation, which we refer as tier 1. finding the epithelial cells, the fibroblasts, the vasculature, the neuroderived crest cells, the mineralized tissue, muscle, and immune cells, which then were divided between myeloid and lymphoid cells. And we could also plot the percentage of the cells across the 70 samples. And from the first look at the data, what we can see is basically, if we just look at the dental pulp data, we can see that it's rich in the neural crest-derived cells and in fibroblasts, while the gingiva and buccal mucosa here in the mucosa are the highly vascularized tissues. And one interesting thing to observe, that we have observed, and it's important for downstream studies, is that different studies using the same niche have vary in the cell recovery, and this really reflects the batch effect, which are caused by different dissociation approaches for the single cell, different sequencing techniques, or even the precise location of the harvesting. So one thing we really need to pay attention is to collect all the metadata that is associated, so that we can check for batch effect factors, right? And this is one of the big concerns for all the data incorporated in the human cell atlas, particularly when we then want to integrate the data sets from different niches in different organs. So now, architecturally, we have two main groups. We'll have the glands, and we'll have the mucosa tissues and the dental pulp, which I'm not going to focus that much on the dental pulp now. So let's just have a better look at the difference between the glands and mucosa, and we move to our tier 2 integration with a higher resolution of annotation. As we can appreciate, again, the vasculature here in green basically is increasingly present in the mucosa, and the immune resident component is also highly present as well, here represented with the blue colors. Of course, there are ductal, acinar, and myoepithelial cells, specifically for the glands. But when we looked at the immune cells by niche, their relative abundance is much lower in the glands. So we really got interested in exploring the immune component in detail. And now, reclustering only the immune cells, plotting them separately for both tissues, and it became clear that the glands are in reach for these plasma cells compared to the mucosa, and the opposite is observed for the mast, the natural killer, and the dendritic cells, which then will be increased in the mucosal tissues. We then move forward to understand what are the molecular signals that regulate the interactions between the structural cells, as the epithelial cells, the fibroblasts, and the vascular, and then with the mucosa, with the immune cells. So basically looking at the interactions between all the structural cells and the immune cells, and then the immune cells between them in glands versus the mucosa, and doing a broad receptor ligand analysis of all these niches and all the cell types has clearly highlighted a hotspot of interactions in the fibroblasts, vasculature, and the immune cells. And then using the multi-niche net tool, basically this is a powerful tool that can make the comparisons of receptor ligand interactions between samples, tissues, and cell types, and produces a huge amount of data that we are still dissecting. But here's a taster of the data that we generated. What happens here is that the arrows actually here on the bottom part will point from the center to the receptor, and they are all colored by cluster, meaning by cell type. And here you have the different cell types from the senders here in the fibroblasts, the structural cells, versus the receivers here, if we look just at the immune cells as receivers. And we can then explore in more detail these structural interactions. And then we can see the top interactions. Also, we are able to understand what are the top interactions between each group. So we have a big number of interactions involving collagen here and here from the fibroblasts, which is not surprising, interacting mostly with the myeloid and the lymphoid cells here on the top that are expressing CD44, CD74, and CXCR4. And then using cell chat to look at the pathways that were being regulated in health from the structural immune cells, we found that the collagens and MMPs are actually essential we know that they are essential in regulating the extracellular matrix organization. And most interestingly, what we see is that there are a certain number of chemokines and interleukins that are involved in the maintenance of these immune cells, and this is constantly happening in the healthy tissue. And this may seem actually surprising to see this upregulation of these chemokines and interleukins in an healthy tissue like the oral mucosa. But if we take into account that the oral tissues are constantly under stress, mechanical and chemical stress, and the pathogens, it basically is a mechanism of rapid response for this tissue. Now, to confirm and actually increase also the power of our findings, we conducted multiplex imaging using the phencycler-fusion platform from Akoya, and we used a panel of 40 markers. And now we are able to validate the presence of all these cell types. And I've mentioned before that at the protein level and with the spatial information. And we have trained cell pools with the different samples of these tissues for an optimal cell segmentation results. So we really need to kind of have a looping system where you train the algorithm with the support and the validation of scientists to be able to have very accurate segmentation, particularly in a tissue that has such a variety of cell types and shapes. And as you can see, we were able to validate the markers for the different type of tissues, here more the structural, with your cytokines, vimentin, vascular, and smooth muscle actin, then the different immune cells and immune cell states. And after we managed to do the cell segmentation, we feed this to a newly developed tacit tool, which is basically an algorithm for cell type assignment and phenotyping, which by giving a set of cell recipes, meaning a list of genes that characterize a cell type, it will set the threshold of fluorescence and predict the cell type. And this was done in collaboration with our colleagues in the Virginia Commonwealth University. Then basically there's a Shiny app that is used as an interface for data visualization, and this allows you for the other downstream analysis, for example, doing the neighboring analysis that can be done using the Delaunay triangulation and a graph-based neighbor analysis. So then we are able to analyze the cell networks connecting neighboring cells within a radius, in our case, which is 55 micrometers, finding what are the most common cell type and its neighborhoods, and by adding up all the number of cells in the neighbor of each cell, we can then build a table of all the total interactions between cells. So the strength of this technique of the spatial proteomics comparing to the single cell, for me, was actually the amount of information that you can extract just from one slide, right? While in our single cell atlas, using 70 samples, we were able to sequence and integrate 250,000 cells here from all the spatial proteomics alone from 18 healthy patients, several of them combined in several slides doing TMAs. We were able to recover more than 1 million cells from these multiplex imaging experiments. So the amount of information, not just in the gene expression, protein expression, but also on the spatial information of this tissue is really incredible, and is the base for this project. But I'm sure that lots of other biological questions will be addressed with the same data set, combining with a single cell. So the first analysis of the structural and immune cell types basically revealed dramatic differences in a cellular and in a metacellular architecture. And using the tacit tool, basically this allowed us to group the cells into any tiered class from the structural and immune cell types, all the way to the cell state that then were annotated for the lower tier cell annotation. For example, if we're looking into activated T cells, which will be a ICOS positive, FOXP3 positive, of course, then with the markers for CD3 T cells, CD4 and CD45, right? So already recognize or allowing you to have this detailed recipe for each cell, then help us do all the cell phenotyping of the tissue. And what we uncovered was the already recognized differences in the immune cell residence that we've seen before from the single cell data, with basically a major differences here in the neutrophils and macrophages in the mucosa comparing to the glands, that we can see here in pink, the mucosa, and then the glands here in blue, where you see the enrichment of these two cell populations. Now, our collaborators also developed another tool called Constellation, basically to classify and analyze the tissue cellular neighborhoods. And so we are able to visualize the cell networks connecting neighboring cells within the radius of 55, and then the results basically show you which are the top 10 interactions, including the different immune cells, that the interactions including the different immune cells and between the structural and immune cells. And to generate an integrated tissue neighborhoods with this constellation, we were able to identify 8 tissue cellular neighborhoods among the mucosa and the glands. And then within the mucosa and the glands, what we see in terms of these neighborhoods is that we see an increase. The fibrovascular hubs are predominant in this tissue, and then the mucosal and fibrovascular tissue cellular networks are more fractured here when compared to the gland. So we hypothesize that this actually may be a reaction again to these barrier stresses that I was discussing before, but also a phenotype that actually supports this more immune cell tropism and an activation in case of constant stress. Now, to show that the toolkit that we have generated, well, that our collaborators have generated, and we are using in this paper, we really decided to apply this to disease samples. And we started by studying one of the most prevalent disease in the oral cavity, which is periodontitis. And for those that are not familiar with this disease, basically in early stage of the disease, what happens is that the gums start to be swollen and red, and then they may start to bleed, and they start to pull away the tooth from the gums, pull away the tooth, and actually what happens is that the bone can be lost and the teeth may be loosened and even fall. And periodontal disease is mostly seen in adults, so we'll see that probably 47 to 50% of adults aged more than 30 or older would form some form of periodontitis disease. And it increases with age. So someone with 65 or older may have, there's a prevalence of 70% in older people. So we integrated only 2 available data sets with both health and disease single-cell RNA sequencing data sets for the periodontitis, giving us more than 80,000 cells just from 24 patients. And again, we performed multiplex immunofluorescence, MERFISH, and single cell, also in this type of disease. So these were the studies published by Drake Williams and the team and colleagues, and Anna Caetano and colleagues. Not surprisingly, what we observe is that there is a great shift on the immune cell populations that are infiltrating this tissue in the periodontitis cases. And this then followed by the analysis pipeline, again, the multi-niche net and cell chart that we used before, we can find that there are altered interactions in disease. And focusing more, and again, on the disease, the interactions between the immune cells and the structural cells, because basically one of the features of the disease is to lose the gum tissue and the structure that basically holds the tooth. We observed an increase in the interactions between these two clusters in the case of the samples with periodontitis. And on a spatial side, here comparing the data sets from the phenoscycler fusion and the MERSCOPE from VisGen, we here using a 300-plex RNA probe. What we could see, and this is really interesting, and this is the kind of information that we will lose with single cell, is the beautiful heterogeneity in the tissue, where you know that in the gingiva, while it is a continuum epithelium, this actually has two sides. It has the oral facing side, so the one that is facing the rest of the mouth, and then you have the tooth facing side, and the tooth facing side is the one that is holding the tooth together. And what we can see is there is an accumulation of bacteria and plaque and inflammation on this region. So we're going to focus on the tooth facing side for now. And what we can observe is that there are some pink cells here. You can see it as well on here. And then there that are the vascular cells, and then you have the blue superbasal cells here from the epithelium, and then here we have also this brown population in this region of the tooth facing, and this brown corresponds to an epithelium superbasal layer at the junction epithelium that is basically adjacent to the tooth, and is this subpopulation that has really a crucial role in supporting the tooth. And by combining the spatial coordinates of the cells and their gene expression using this loop on the MERSCOPE data, allow us to specifically identify not only what are the cells, but actually what are the pathways that are upregulated in period disease, and what are the signaling pathways that we will need to use to target for the development of future therapies. And that's indeed what we are analyzing at the moment, and looking into data sets of different drugs to be able, and comparing these with our targets to be able to find possible drugs that could be used to target these specific inflamed pathways. But again, as I was telling you, a very important part of our work is that we are developing to contribute to the field and give others the opportunity to access our data. And we are then working to uploading all these single cell atlas into Cell by Gene. This actually, some of them are already online. And this is how it looks like at the moment. So basically, it's a platform provided by Canon Zuckerberg Initiative, and is a tool that helps scientists to find, to download, to explore, to analyze and annotate the data of published single cell data sets, and now also with spatial data sets. And it includes several ways you can engage with the single cell data. So one can check for the expression of your favorite gene, or select for different tissues, different conditions, do a differential gene expression analysis, and much more. So some of the data that are presented here is already in Cell by Gene for all the community to explore. And why is this important, if not essential for us, is because we want to make sure that most of the data generated is available, so that other labs can use, and ensure that every person, an untrained PhD student, a clinical researcher, or even my mom that is not even versed in bioinformatics, can access the data and the tools for some initial basic analysis to explore new hypotheses on their projects. And now this brings me to one of the main biological questions that we are exploring in my lab, which is, have you ever noticed that when you bite your tongue or your cheek, it hurts? You bleed a lot, but it heals much faster than the skin. And most importantly, it leaves no scar. And it is well known that when compared to skin, the oral mucosa wounds heal much faster with little or no scar formation. And that is what makes the oral mucosa so special and what made me so interested in studying this tissue. The oral mucosa, basically, the wounds reepithelize faster, mainly due to differences in keratinocytes and in fibroblast proliferation, together with lower levels of inflammation and angiogenesis when compared to wounds in the skin. And in a way, it is said that, and we also know this, that skin from babies and from children actually scar also much quicker, right? And again, the wounds are not as prominent as a wound in an adult. And what is said often is that the oral mucosa actually behaves like a fetal or a neonatal wound in the skin, right? Kind of being able to regenerate fully. However, while we already know so much about the comparison between these two tissues, there's still a lot to be explored concerning what are the mechanisms behind this scarless potential and what can we learn from the oral mucosa to then apply to skin wounds. And today, the second project that I'm presenting is really focusing on the role of fibroblasts in these processes and how we are using the human cell atlas data that I've just presented to answer these questions. And this is a project led by a very talented PhD student in my lab, Diana Pereira. So, in parallel with the human oral atlas project, we have recently generated a single-cell RNA sequencing atlas of the mouse, neonate, and adult, of the different tissues of the oral mucosa using the 10x platform. So we are able to capture all the cell types that make up the different tissues of the oral mucosa. So here you can see all the annotation, and we are also used immunofluorescence to validate all the tissues that we recovered from the single cell. And we first dived into understanding how the fibroblasts, because that's one of the main questions that we want to address, is how these fibroblasts are different between these two tissues to support this wound healing, how the fibroblasts of the oral mucosa are contributing to the high regeneration potential of this tissue when compared to skin. So for that, we integrated the fibroblasts of the oral tissues with the skin fibroblasts from published datasets, in this case from our collaborator, Maria Kasper at Karolinska Institute, and other published datasets. So we can identify, very interestingly, that in neonatal there is an overlap between the fibroblasts of the oral and the skin data sets. However, if you look at the adult data set, sorry, there is a mislabeling here. If you look at the neonatal, what we can see is that there are shared fibroblast subpopulations between neonatal and adult, all right? Between neonatal of the oral and the skin. However, if we look at the adult tissues, please ignore these two labels. If you just look at adult tissues, what we can see is that as the skin and the oral mucosa evolve, what we can see is that the fibroblasts don't integrate so well. What we would see is that there is a cluster, there is indeed shared between skin and oral fibroblasts, but then there is a skin-specific cluster, and the skin-specific cluster has fibroblasts that have a different identity. And interestingly, one of the markers that we've seen that is enriched in the adult oral mucosa and some of the skin fibroblasts is expressing mFAB5. But this is not expressed in the cluster that is specific just for the skin. So mFAB5 is actually a gene that encodes for a microfibrillar associated glycoprotein 2, and this is known as a component of the elastic fibers, promoting cell migration, and could be an interesting molecule to explore further. So we then did some spatial analysis to see where it is located, and indeed is located in the most papillary fibroblasts of the skin, so the upper fibroblasts of the skin, and then in the oral mucosal tissues. And this is something that we are exploring further to look at other interesting markers. But for us, what is then important is to understand the functionality of this fibroblast. I'm just going to briefly show you how we're going to analyze and uncover the different functions and regenerative potential of this fibroblast in vitro. And so what we have observed by doing 2D and 3D in vitro assays is that the oral fibroblasts do have an increased proliferation and migration capacity. The buccal fibroblasts contract faster than the skin fibroblasts, and we know the oral fibroblasts are better in supporting the epithelial growth here by doing an organoid co-culture of the epithelial cells, both from skin and oral mucosa, with the different subpopulations of fibroblasts that I just described. And we could see that these oral fibroblasts do support better the epithelial growth than the skin fibroblasts. And so by uncovering this heterogeneity in the oral mucosa using single cell, and then doing this functional analysis of the different subpopulations using the markers that we uncovered, we are then able to, in vitro, but ultimately in vivo as well, be able to understand how the different subpopulations are actually functioning, not just what genes they are expressing, but what are their actual functions in the tissue regeneration to then be able to apply for physiotherapies. But another question that we're really interested is in this cross-species analysis, right? Trying to understand, are these subpopulations conserved and the markers between them conserved between mouse and human? So what we're doing at the moment is comparing this annotation of the fibroblasts between the two species, their spatial location, their heterogeneity across the niches, and just to give you a flavor of our first analysis on the single cell, not on the spatial yet for the mouse, but it's coming, is actually by integrating the integration of both the mouse fibroblasts that I just presented, and here just in adult, with the human fibroblasts from the oral cavity that we got from the human cell atlas that I presented in the first part of my talk, with the skin fibroblasts from other data sets in the human cell atlas for the skin, we were able to cluster the different subpopulations and looking at the distribution of the fibroblast subtypes by tissue. We see, for instance, that the mouse lacks an immune response specific fibroblast cluster. And interestingly, the mouse skin is really enriched in DCM production fibroblasts, while the oral tissues have more what we would call the tissue development related fibroblasts. So we really want to now take a step further to look at the different markers between those, and now with the data sets that we have available for the human and that we are finalizing for the mouse, being able to map these subpopulations and look how they are working and communicating in a spatial architecture. And with that, I would like to thank all the people involved in these different projects. This is a real multidisciplinary work that wouldn't have been possible without the help of several people, to whom I'm very grateful. First of all, to our collaborators. The Human Cell Atlas has been a very productive, a very fun and team science work that has been done in close collaboration with the team of Kevin Bird. And I would like to acknowledge also our Genome Center and Single Cell Genomics at UCL, and our collaborators at the Skin Atlas, Maria Kasper, and the team at the VCU, and Mike, that has been helping us with the bioinformatic analysis, and of course the funding that helped me set up the lab, develop all these projects in the lab, and the fellowships for my lab members. And last but not least, thank all the members of my team, a group of very talented students and scientists that are super dedicated and enthusiastic, and a real pleasure to work with. So thank you all for your attention, and I'm open to your questions.
Dr. Sequeira, thank you so much for this informational presentation, and welcome back on camera here for our live Q&A portion of our webinar. Very good. Let's get you back on camera.
Did I stop sharing?
You stopped sharing. Yes, we're all good, except for some reason your camera is not showing. It's showing a black box. But I'll give you a second to troubleshoot that. But real quick, I do want to let our audience know we do have some interesting questions that have already come in. And to those of you who have not submitted any questions yet, we encourage you to do so now. Simply type them into the Ask a Question box, click Submit, and we will go through all your questions and answer as many as we have time for. Let's see. Dr. Sequeira, I'm just going to have you real quick check your device settings, and it's possible that it changed cameras when you were sharing your screen. Your device settings are at the top right of your screen. It's a little cog wheel or a gear icon. You see that? And that gives you options to select your camera. I want to make sure we can hear you as well, just in case you changed your mic. If it's helpful, maybe do a quick refresh of your browser and reconnect. That might work. We want to make sure that we can see you and hear you. All right. Well, to our audience, thanks for your patience here. We're just going to get a quick reconnection with Dr. Sequeira. And let's see. There we go. Excellent. Perfect. Sometimes we all just need that little refresh. Well, excellent. So Dr. Sequeira, we've got some great questions. I'm just going to dive in here. So our first question, what are the changes that you observed between the mouse and the human models?
Well, first of all, a lot of the markers were not conserved. And this is really interesting because it makes us understand that I spent, I don't know, 15 years of my career working on mouse models only. And a lot of the basic research that we've done in mouse with different markers, then it was super important to help us understand cellular behavior, and particularly on stem cell regeneration and tissue regeneration. But a lot of the markers are not conserved. So if we then want to translate this into humans, this is much more difficult. So being able to have access to much more information out of the human samples with these multiomics platforms is really a great advantage, and frankly at the moment it's super exciting to be working on this field, to be able to compare both models. But we need to be very careful when trying to extrapolate all the markers and findings in cell types that were observed into mouse models into human, because it's not as straightforward. So doing this dual analysis is always important.
Excellent, thank you. Now, what kind of immune cell phenotypes are involved in mucosal cavity for wound healing?
So what you'll see is, first of all, in the oral mucosa, you see much less vascularization, right? That in the much less inflammation there in the skin. And then there is also not different cell types that play a role, but also a delay on the timings for the skin. So if we compare the wound healing in the skin, for example, giving you an example of a 2 to 4 millimeters wound in the mouse, it may take 2 to 3 weeks to resolve for the skin, while in the oral mucosa, in 10 days, in 4 days, you already have a full reepithelialization, and in 10 days, is completely closed. And all the cell types, if you look at the dynamics of the immune cells, are different between one and the other. So you have the platelet cells that really play an important role. The different other different immune cells, such as the T cells, but also a lot of the macrophages, and in the oral mucosa, you really see also, particularly in the gingiva, an increase of the neutrophils. So they play different roles, and actually the timing of the contribution of cells is slightly delayed for the skin comparing to the oral.
Thank you, Dr. Sequeira. Now, here's an interesting question. You mentioned having samples from a diverse ancestry. How are you achieving this, and why is this important?
Well, if you think about considering different diseases, for example, prostate cancer is known to be with a higher incidence in African descendant ancestry, or for example, keloids in the skin, this highly fibrotic tissue in the skin that usually appears in the ear lobe, also has a higher incidence on another, on a skin type associated with African descendant patients. And there, like this, there's a lot of other diseases that are differently affected with different ancestry background. So we need to understand the diversity of these different backgrounds. While the Genomes Project was very much focused on whites, probably North American donors, volunteers, we really want to make sure that the samples and that are contributing to the Human Cell Atlas have really capture this global diversity that we have, so that we are not actually finding possible targets that are going to be useful for a subset of our world population, but actually can be beneficial to everyone. So being able to study also the diversity on a global scale, it's important when we then take these studies, these data sets, to downstream analysis for human health.
Thank you for elaborating on that. Now, do you plan to increase the plex number for future experiments?
Yes, we always want more, right? Definitely for the spatial transcriptomics, we are already doing it, and using technology is also evolving. I was presenting you data that was probably generated 3 years ago, and we are now finalizing the analysis. I think here more the bottleneck is the analysis than the technology is evolving itself. So yes, for the spatial transcriptomics, we are increasing. For the multiplex imaging, it can be increased. It's just a matter of, and I think there's proof of concept from Akoya that you can use 100 markers. And what we are also doing, and is like one of the main focus of my lab now, also with our interest in mouse models, is also developing panels for mouse tissues to be able to study also in a spatial level our mouse models. So yes, that's in the pipeline.
Great news. Now, our next question. Hi, Inês. Awesome talk. Thank you so much. I was wondering, will this ever be pushed towards investigating oral carcinogenesis as well?
Well, that's also one of the projects that we are developing in the lab, and I'll be really happy to collaborate further or explore getting samples from other places to be able to expand our cohort. We are very privileged here in East London with a cool, interesting community with a lot of Indian and Pakistani patients that develop a lot of oral cancer, particularly from chewing tobacco. But if we have access to samples from other teams across the world, this is really something that we'd like to explore, other backgrounds and other risk factors on cancer development. So yes, we are expanding on this, and if it's something that is of interest for you, I think it was Martina that posted the question, it's something that clearly I would like to expand more, so do get in touch.
Yes, and Martina, you can always come back and submit your questions during the on-demand period. We do have your email address, and the answers can be sent to that email address. So thank you for your question. Next question, are your fibroblasts equivalent to stem cells?
I don't think so. We are looking into differentiated fibroblasts here that we labeled in an adult. We are not really looking into, let's say, mesenchymal stem cells. But these fibroblasts that I was defining with a more regenerative potential are actually better supporting the stem cells of the epithelium, kind of sending signals like KGF, keratinocyte growth factors, that really support the proliferation of the basal cells, so the stem cells that are in the epithelium, to support their cell division and the repithelialization of the tissue and the wound closure. So when we say that they are more pro-regenerative, I think that's what you're trying to do. The question that you're asking is basically that they secrete factors that are supporting better the epithelialization of the tissue. And this is kind of something that has also been functionally analyzed in human by the lab of Fiona Watt, where you have when you co-culture the papillary fibroblasts of human with epithelial cells in 3D organotypic cultures, what you see is that indeed these fibroblasts are supporting better the formation of the epithelial tissue and the proliferation of these cells, leading into a thicker epidermis when you compare, for example, with reticulary fibroblasts or lower reticulary fibroblasts. So they are not considered stem cells per se, but they are supporting the proliferation of the stem cells.
So Dr. Sequeira, how do you determine the number of samples needed for a spatial transcriptomic study? For example, you mentioned 18 subjects for oral mucosa study.
It's very difficult. It really depends on the heterogeneity across. Here we were just talking about healthy individuals, right? And this is a question, and grand panels have asked me this question before, particularly if we think about cancer that has, for example, you don't know, if you have 20 tumors from different patients carrying different mutations, well, here you're not looking into an homogeneous population, right? Because we need to take into account the different genomic changes. So this is something that it's really difficult to answer. The advice I have, if you're trying to do your power calculations to decide the number of samples, is focus in groups that are slightly homogeneous to start with, to be able to find the main changes, and this gives you the statistical power for your analysis. And remind yourself that you can actually optimize the costs, because ultimately also the costs is something that will really limit the number of samples that you can do, but you can optimize by doing TMAs and putting several samples on the same slide, and also reduces the variability across different stainings. This is something that you could do to increase the power of your study. But yeah, I would love to be able to do more, but we need to, in healthy tissues, probably it's easier to control this variability. Mouse much easier than human, for sure. But in in cancer research, you also need to take into account the intrinsic heterogeneity across different tumors.
Thank you. Our next question, what are the interactions involved between fibroblast and immune cells, macrophages, in oral cancer or wound healing?
Sorry, again. Well, we know very much that fibroblasts well, there's a lot of communication between them, that's for sure. That the immune cells are, for example, in wound healing, the more inflammation you have, well, depending on the type of inflammation, but they can really lead into these fibroblasts being depositing more extracellular matrix, and these extracellular matrix at the beginning are important for the closure of the wound, but then can become pathological, in a sense, can actually create a bigger scar. So these increase in inflammation can also be something that it's not ideal in the wound closure. But for the macrophages itself, definitely they are also important to then help clearing the tissue as the wound bed is remodeling.
Excellent, thank you. Our next question: what cell line is currently available to grow in your cultures?
I'm not sure I understand the question. There are several cell lines that are available to grow in culture. The culture I presented you is basically from primary cells, so it's not a cell line. But we tested with tumor cells. However, the organoids were not as nice as primary tumors, just because the tumor cells were not growing in a very organized manner to form the organoids.
Very good, thank you. Our next question: how are the oral immune systems different from peripheral organ systems? And is there any gender differences that you've noticed?
I would love to know, but I cannot answer that question. I really, it's something that we want to explore more, but again, not enough statistical power to look into that. But I would love to do that. And I know that in skin, they are really looking into that in detail, particularly thinking about the hair follicles and all the hormonal changes and how they are controlling hair growth and tissue revitalization, etc. So this is something that questions that we'd love to answer, but we don't have answers yet.
Well, hopefully we'll continue that research, and that could be another webinar. So, Dr. Sequeira, have you analyzed oral cancer samples using spatial techniques?
Yes, and that will be again for another talk.
Excellent. Stay tuned. Excellent. We're coming close to the end of our webinar, but I want to squeeze in just a few more questions. In your human model, are you performing scRNA-seq of normal and activated oral mucosa in a wound healing situation? And which and how many subpopulation of fibroblasts can you observe? Finally, are there subpopulations changing in normal and wound healing oral mucosa?
That's so interesting. You've probably been reviewing my latest work. Yes, it's really something that we are interested, and in terms of subpopulations, yeah, there's more inflamed subpopulations that are seen, and there's a cool work also that has been done on skin. Looking into the different subpopulations of fibroblasts in the wounds in skin. I really suggest that you have a look. I think I've seen a presentation from the lab of Klein and Michael Longlaker on that, and it's stuff that has really been pioneering also these analysis of the subpopulations of fibroblasts in wound healing. But yeah, we're definitely interested on that.
Excellent. And Dr. Sequeira, we're going to wrap with this final question, but I do want to remind our audience that those questions that we are unable to get to due to our time constraints here, and additionally those that are submitted during our on-demand period, they will be addressed via the email address you provided at the time of registration. So don't stop submitting those questions. Come back, watch again, submit more. We'll get those questions answered to you. So our final question of the day, Dr. Sequeira, have you tried to use these fibroblasts to shorten the time of healing of a cut-in skin wound?
Well, following up what you're saying, this is something that has been beautifully done in even humans, I think. No, actually they were not grafting. But this is something that has been done by other groups. We're also interested, but now looking into the different subpopulations. And we have in-house expertise to do this grafting of the fibroblasts, and frankly it's how we want to go forward, particularly considering this for new strategies for wound healing to be applying in humans. Thank you for the question. Is really how the field and our lab is moving to. The email is actually on the slide, so you can have both the slides. So someone was posting a question about asking for my email ID, so you can see my email and the website of my lab on the last slide, so feel free to get in touch. And we have positions open as well if someone got interested in our research.
Very good. Yes, we do have that slide in the top left of your screen. You can expand that slide so that you can see it bigger. But Dr. Sequeira, thank you so much for sharing your expertise with us and in sharing the different and complementary outputs of single cell and spatialomics, and for helping our audience understand the cellular heterogeneity of skin and oral tissues. Do you have any final comments for our audience before we close?
I think, well, first of all, thank you for all the fantastic questions. And I'm happy to get in touch. Thank you for giving me the opportunity to present this webinar, and whatever research you're going to be doing, I think combining different tools is really kind of key to taking the most out of your samples. So if you have the chance to and the funds to do it, is really something that I advise moving forward. So good luck also with all your work, and please do stay in touch.
Excellent point. And thank you again, Dr. Sequeira. I also want to thank our audience for joining us today and for their interesting questions. We had so many great questions coming in. Again, don't fret, those questions will get answered via your email address you provided. I do want to thank LabRoots for hosting today's webinar, and to our sponsor, Akoya Biosciences, for sponsoring today's webinar. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. I do want to bring your attention to a survey that will pop up in the middle of your screen as soon as we close. We encourage your participation. It does help Akoya improve their webinar program. We'd love to hear from you. Thank you again for joining us, and thank you again, Dr. Sequeira. Until next time, everyone, take care and have a great day. Bye-bye.